Hello everybody and welcome back to my YouTube channel. And in today's video, I am going to be discussing about the different domain specific research and how can we have a start to end work plan, right? So when we talk about the pharmacy or medical profession in general, we have a variety or a plethora of domains that we can choose from: pharmacology, pharmaceutics, pharmaceutical chemistry, analysis, medical devices, clinical research, pharmacy practice, regulatory affairs, and then after all this, pharmaceutical marketing. So this video will encompass a complete roadmap of how to brainstorm for a research up to the execution of that research. So these are the different steps or the common steps you can tell that any domain, कोई भी domain में आप जाओ, these steps have to be followed. Firstly, a thorough literature search, primary as well as secondary literature review is very important, right from your journal selection up to the article reading. Then you need to identify the gap. So rational and gap analysis is very important. Missing क्या है? Why will, if you even if you're writing any article, or क्यों लोग पढ़ेंगे? Why will people read your article? There has to be something unique, something additional, extra. Then you can, if you are from some pharmaceutical marketing point of view, you can have a commercialization aspect or the trend analysis or market insights. That just skin whitening products. If you are preparing any pharmaceutical cognosy and Sutix का combination, any cosmetic preparation, कौन सा cosmetic ज़्यादा is, can you prepare kojic acid formulation for skin whitening? Can you prepare arbutin or something like that? Then we have experimental resources and setup. So wherever you're working, you need to have an idea कि हाँ हमारे पास ये चीजें हैं, ये setup है, these all equipments or instruments are present in the premises, in the facility. Designing a experimental design and protocol, that is very important because वो आपको एक timeline देगा कि हाँ you need to complete this within this span of time, this is to be done after this, तो एक sequence, ये cascade होगा, right? Then collating the results in the discussions that you have obtained. Finally, publications and patent. This is a very important step. Mostly लोग research करते हैं बस उसे publish नहीं करते. They don't reach up to that step. They think that हाँ degree मिल गई or whatever is the objective present कर दिया खत्म. No. And after, even after publication, make sure that you translate it into different kinds of like seminars where there are so many different opportunities where you can present your work, right? Hackathons होते हैं, conferences होते हैं. Make sure आप अपना जो काम किया है उसे लोगों के सामने दिखाए. Winning of course should be a second priority, but first आपको knowledge को disseminate करना है. That is very important. So these are the different publications that I have on my name currently up till now, and that is the reason I'll be covering most of the topics because I have somehow or the other touched few of the topics, right from your informatics to your sutics to your ecology to your cognosy. So maybe it would be of a little help. And these are the different glimpses or the pictorial representation of what all work I have done till now. So this is a anti-acne face scrub. This is also a foam test, saponins. This is spreadability. This is a test for anti-inflammatory, denaturation, albumin, protein denaturation test. This is a cream. This is a cystone test for nephrolithiasis activity, anti-kidney stone. These are the different images of zone of inhibition or antimicrobial studies. These are the docking scores. So I have also performed docking and all. I've used Pymol, I've used Schrodinger MOE. So different structural maps, binding scores, different ligand interactions, and different pocket sites or co-crystals I have been identifying. Then these are different kinds of plots that I've designed using DOE, design of experiments. This is the contour plot. Then IVD, in vitro drug release, CDR, cumulative drug release. These are different drug diffusion studies performed using France diffusion cell. Then the stability testing using the stability chamber. These are the microwaves, green chemistry. So when we talk about the green chemistry, that is also very important. Then the different kinds of evaluation parameters for analysis like HPTLC, TLC, Brookfield viscometer. Then this was an image of thymol from carom leaves of different kinds of extraction process of oxidation. This is again stability, tap density, bulk density for particle size. Then again, these are antimicrobial, and these are the images of the gummies that were made. These are the SEM10 images of the niosomes. So we had prepared niosomes as well. Then this is a Sudan red test for volatile oils. These are the different, again, pictorial representations of the different studies that we have carried out. Again, these are the HPTLC images for mulberry and chia seeds. So we have prepared an ice cream using this mulberry and chia seeds for obesity. And these are the different images of how we obtained mulberry, powdered it, chia seeds, powdered it, and then finally created a paste. And this was done by my students, actually, by my under my guidance. So I'm really thankful to them, Shruti and the team, for their contribution in this work. This is a pH meter, ash value, extractive value, flowability test, the different case spreadability, and this is the image of TLC, again column chromatography. Then these are the, again, sieving and size reduction, separation and all. This was the ice cream. This is another ice cream that we had made, or not ice cream, this was, I think so, yeah, this was candy sticks that were made. So it was freeze dried, freezed, and then and these are the different extracts that were used. And these are the microscopic examination images. These are the, again, gummies that were made. These were herbal gummies for depression. um, uh, and then these are again image similar, all of these are images, corneal permeability test, and these are meswak buccal film images. Then this is a time point assay, and then Brookfield viscometer, magnetic stirrer. So these were the different instruments on which I've worked. So that is why in this video I'll be discussing about the different concepts that we can do. Then speaking about the publications, I have 4 Scopus Index publications and 5 articles are under revision on the Scopus. But the total number of publications are 16, Scopus as well as non-Scopus. Number of poster presentations are 11 till now, and under my guidance I have my students who have carried out around 12 poster presentations, and these are the images of the different articles that I have published, and book chapters, short communication, reviews, research, based on male contraceptives and all, liquid solids, moringa, and polyherbal instant premix, and anti-acne face scrub, effervescent tablets, hepatoprotective models. So these are the different kinds of articles that I have published in the Scopus Index journals, that is from Indian Drugs and IDMA, IJPSN, and European Journal of Hematology. So you can have a look at this as well. I'll be attaching the links for all of these in the description section so that you can have an idea, because in this there was a really good data, because there was spinning technique, and there are different wound healing reepithelialization data, a lot of microscopic data, so different diabetes induction data and all. Now when I talk about a publication, a manuscript can be published in variety of ways. Most of the problem कहाँ आती है कि हमें audience को पता ही नहीं होता है review and research के अलावा और क्या होता है. There is original investigations, clinical case reports, commentaries, special issues, LTR, which is letter to editor, short communication. मेरा एक LTR is also being published. I will attach that as well, so for your reference, because most of the journals do mail you that they need a letter to the editor. So they take out certain kind of notifications. Then there is short communication, there's a review, there's a research, there's a mini review, there is a meta-analysis, network meta-analysis, and there's a book chapter. So if you are not able to write a review, or suppose आपका research आप कर रहे हो but you don't have that significant data and it's stopped, you can publish it as a short communication. That will work. Meta-analysis, if you have done bibliometry analysis or anything, if you have any survey data or any kind of data that can link multiple things, of course you must try for that. You must go ahead. Now, when I speak about an experimental study, so the first subject that we are going to cover is pharmaceutics. So in pharmaceutics, the first or anything, identifying the gap, problem, literature review kariye. And you can have an idea of the different salt forms, ester forms if you want to go for bioequivalence, or generic, complex generics, branded generics, or if you're feeling for going for an inhalation, you can design any kind of product, right? From your novel drug delivery system, liposome, niosome, ethosomes, dendrimers, transferosome, to your conventional drug delivery system, tablet, capsules, parenteral, aerosols, inhalational products. And you can even integrate microneedles in the transdermal patches and all. So you need a regulatory ethical and considerations. You can have an idea about the FDI guidelines, the ICH guidelines, the if there are any animal studies involved, in the institutional ethics committee, animal ethics committee. Then you need to identify whether the raw material, the API is available, because mostly we get gift samples. So for gift samples that we get, we have to show the COA, certificate of analysis, कि हाँ भाई ये जो drug मैंने अपनी study में use किया है, that drug is having this much % of purity. right? Then you have to do a method development. So it's a important process of the method development, because in a preliminary formulation, the HPLC method needs to be developed and validated, right from your LOD, LOQ, robustness, ruggedness, accuracy, precision, linearity, to all those things are required for the validation purpose. And then we need to identify the resources budget. So set up a design, like you can use a DOE, design of experiments, and quality by design. So you can have a TPP, target product profile, and QTPP, quality target product profile. So in this you need to identify the various CQA, CMA, CPPs, critical quality attributes, critical material attributes, critical process parameters. Then you need to identify if there are any confounding variables, if there are any, any extraneous factors that may affect. So then you need to go for the preform studies, where you have thermodynamics, pH, solubility studies, compatibility studies. So drug excipient compatibility can be checked via the FTIR, Fourier transform rate. You can also check DSC, TGA data. You can identify if you're preparing an amorphous solid dispersion. You can have an PXRD, powder X-ray diffraction as well. Then you need to identify the polymorphism. You can identify polymorphism as well, pseudopolymorphism using hot stage microscopy, stereo microscopies. There are some type of microscopies as well available. So then once it's done, optimization is done, you can have a DOE for the, box Benkin design, CCD, central composite design, and all for optimization. And then finally you can have analytical techniques like UV, HPLC, and all. And then according to ICHQ2 for analytical validation, you have specificity and all those different parameters involved. Once the formulation is formed, physicochemical characterization like the assay, the dissolution studies, like dissolution is very important. BCS class, which it belongs to, BDDS class, which it belongs to, the pH, the viscosity, which viscometer you're using, Brookfield, cup and bob, cone and plate, particle size, so you can have zeta sizer, Malvern analyzer as well. So you can identify with the help of those particular instruments. Then France diffusion cell or dialysis cell or any USP apparatus cell for permeability studies, like if you can perform CACO2 permeability assay as well. Then after this, you can also perform ex vivo studies, like you can design a simulated fluid, simulated intestinal fluid SIF, simulated gastric fluid SGF, simulated nasal fluid SNF. There are so many different simulated corneal fluid or lacrimal fluid. There are so many different fluids that you can design and create an artificial medium like salivary amylase. If you're creating any formulation that needs to be, chewing gum or anything that is buccal formulation or anything, you need to identify its activity in the salivary amylase as well, right? So those are activities and studies you can perform. Then for pilot plant and scale up synthesis, the process development, you can optimize the various manufacturing processes like granulation, coding, lyophilization. And then finally you can go move on to animal studies. I'll have already covered a lot of parts of animal studies in my previous videos as well, so you can go and check them out. And also in today's part we will be covering in pharmacology, in silico, in vitro, and in vivo techniques. Now next up I'll be talking about the pharmaceutical management. So marketing management is very, very important. First you need to identify the research questions, whether are you targeting the STP, segmentation, targeting, positioning, are you targeting the marketing, whether it's a differentiated, undifferentiated, niche marketing? Are you targeting the promotion strategies, online, offline? Are you targeting the marketing strategies, skimming, penetration, psychological? Are you targeting the patent strategies like the blanket patenting and all? Are you targeting the PLC, product life cycle? And are you targeting any market entry, NPL, new product launch? So this will help you to establish a good particular, basis for creating a particular research. Then literature review can perform using market research, case reports, case studies, and as so many different gaps can be identified. Then you need to choose a particular research methodology, whether it's going to be based on statistical modeling, data analysis, surveys, case reports. Are you going to have an expert interviews? And research can be qualitative or quantitative. So it depends upon you whether you are trying for qualitative or quantitative. Then you need to identify, formulate a hypothesis, like if it's related to regulatory approval for a particular drug launch, that can be there. Then you can also design a framework like SWOT analysis, strength, weakness, opportunity, threats, special analysis, political, economical, social, technological, legal, those kind, environmental, Porter's five forces, bargaining power, buyers, suppliers, and industrial rivalry and all. So then you need to select a particular target audience, whether you're going for a global marketing or rural marketing, right? Then you need to identify the various conflicts, potential conflicts, or competitors. So competitor analysis is very important. That is a growing field nowadays, because competitive. We need to have an idea about the various trends that are currently going on. Then the data collection. So you can have a data, primary data as well as secondary data. Primary data मतलब basically कोई interview, survey करके you get, right? On field or on site data. Secondary data means कोई reports published है, कोई competitor statements है, financial statements. Those are classified as secondary data. Then you need to perform the market analysis, like the different regulatory bodies on the product development. So you must have a very close eye on the FDA warnings or on the different competitors, or you can just visit the website and have an idea about the different financials. You can get there, all the EBITDA, revenue, or COGS, cost of goods sold, and all. You can have an idea about those as well. And the key management strategies like the DM drug manufacturing, the different intermediaries or distribution channels, like you have stockist, super stockist, wholesaler, retailer, distributor, inventory management, then navigating the different pricing approvals, because pricing is very important marketing point. So and there's NPPA pricing, National Pharmaceutical Pricing Authority, that, and then there is uh, DPCO, Drug Price Control Order, and all related to that. Then you have a business case development. So in this you can include the different return on investment or your risk assessment, that also you can involve sustainability, because nowadays most of the companies focusing on CSR, ESG, environmental, social governance, corporate governance, and all of these things. So you can have an idea about those as well. Next up, we have pharmaceutical chemistry. So in chemistry, we can have green synthesis, we have peptide chemistry, analytical chemistry, inorganic chemistry, organic chemistry. So you can have a project planning right from your equipments to your reagents, because reaction lagana hai to reagents चाहिए, reflux, condenser. If it's a diazotization, then we need good amount of temperature. And mostly of the studies that you do, you need to identify target identification. So QSAR studies, like quantitative structural activity relationship, HTS, high throughput screening, and docking. So you can have different docking softwares. You have so many different Autodoc and PyMol and so many RASMol. There are so many different softwares, Vaccegen and all for vaccines. So you can just use these particular tools and software for predictive analysis. Then there is lead compound identification. So for in silico screening, for natural product based isolation studies, ADMET analysis, you can have these particular things. You can also perform purification, because in analysis we do impurity profiling and metabolite profiling. So similarly, or fingerprinting. Similarly, you can do here the different kinds of analysis that are required: purification techniques, crystallization, column chromatography, and in vitro studies you can perform IC50 value, inhibitory concentration, EC50 value, effective concentration. You can identify the binding affinity, you can have a target score, binding score, grading score. You get different scoring techniques. And then finally toxicity testing for those identified hit molecule. Then moving on to the next subject, which is pharmacognosy and natural products. So the first steps remains the same: literature review, gap analysis, regulatory approvals. Now what changes is the collection and plant material selections. So you have voucher specimens, right? Some people call it herbarium as well. So there is a particular voucher specimen that is recognized for future reference. And for botanical authentication, we need a lot of different approvals as well. So we need to identify with a different extraction process. Firstly, maceration, percolation, digestion, decoction, infusion, microwave assisted, countercurrent, which kind of extraction, supercritical fluid you want. Then preliminary phytochemical testing, like if it's having primary metabolites, carbohydrates, protein, lipids, a different test. We have Molish, Benedict, Barford, Selivanov, Felling, Tollin. You have different tests for carbohydrates and proteins. For lipids also you have Lieberman, Burkhardt, Salco, Whiskey, and all. For similarly for secondary metabolites, we have tannin, glycolide, glycoside, flavonide, volatile oil, resins. So for them you can perform different testing like Shinoda, Mayer, Wagner, bond taggers, and all these kind of testing procedures, right? So in this you can have a sample dry preparation. So for this you have microscopic analysis as well as macroscopic analysis. For organoleptic, color, odor, taste, and all is suffice. For microscopic and all, we and different kinds of analysis: physical, chemical, biological. So a particular analysis has different domains: physical analysis, chemical analysis, biological analysis, phytochemical analysis, microscopic analysis. So vein islet number, palisade ratio, stomatal index, ash value, lycopodium spore method, extractive value, LOD, loss on drying. These are different kinds of experimentation you can perform when you're performing for partition coefficient, because partition fractions and extracts is very important. So HEA, hexane ethyl acetate, or butane. There are different mobile phases that can be used for partitioning. TLC सबसे मेरी major factor है TLC. We need to match the RF value. So that is, and for optimization of extraction, you can even incorporate DOE into your process. So design of experiments can give you different variables. It can be a 2 factorial design, 3 factorial design, or 2 square, 2 cube, 3 square, 3 cube, depending on the levels and factors you have in your experimentation. So chromatographic techniques and characterization using MS spectroscopy, NMR spectroscopy. Then you have different chemometric tools as well for which data can be analyzed, like HCA, PCA. Then you can perform in vitro bioassays like DPPH, diphenyl phenyl hydrozonium assay, FRAP assay, fluorescence regulatory after fluorescence recovery after photo bleaching assay. There are different assays that you can perform for antioxidant activities. There's a TRAP assay, total radical antioxidant power, free radical antioxidant power. There are different FRET assay, fluorescence resonance energy transfer assay that you can do. Then for antimicrobial activities, the best is disk diffusion assay, right? Antimicrobial, there's a cylindrical plate method, there's turbidimetry method for assays and antimicrobial activities where you check the ZOI zone of inhibition. And enzyme inhibition kinetics also you can do, like amylase, um, tyrosinase, or any enzyme you tell, like acetylcholine. If you're working on Alzheimer's, there's acetylcholinesterase. Then in vitro toxicity assays you can do by performing MTT, XTT, MTS, XTS, right? So these are different dyes so that are responsible for cell proliferation, cell viability assays. So you can perform those assays as well. And if you're performing toxicological OECD guidelines are very important. Once you have all of this, you have data, so you need to compile that data and perform a statistical analysis using ANOVA or t-test, depending on the number of groups that you have. And then finally go for product optimization, dissemination of this information, manuscript preparation, and on a higher scale with a greatest amount of sample quality control, QA/QC testing. The next subject that we'll be covering now is medical devices. So medical devices requires a integration of biomedical engineering, biomaterials, and your pharmacology, because there are different biomarkers that you can estimate, or even for tracking, like medical device for tracking an Alzheimer patient, medical device for tracking the cortisol levels for stress, medical device for measurement of oxygen, SpO2, oximeter, for glucometer, blood glucose level, right? So there are different medical devices that are available. These are the different medical devices. Besides this, you also have objective medical devices, like you have stents, balloons, heart stents, prosthetics, implants, orthopedics. So depending upon the limbs, upper limbs, forelimbs and hind limbs के अलग-अलग medical devices होते हैं. So based on those particular parts, you can design a particular medical device. Then there's a 3D bioprinting or an organoid culture now being very famous for, like 3D bioprinting technique can be useful, where the fluid dynamics and all you can, just control and you can control those variables. Then you need to identify whether it's a mechanical device essentially. Like now there are tattoos. There are tattoo electrodes for measuring the cardiovascular risk, right? So these are novel researches that are happening currently that you must know. Tattoos and all that are being updated recently. So you can also perform the biocompatibility assessment. That is very, very important. Then when I talk about the different regulatory aspects, so medical device have different categories, my class 1, class 2, class 3, class 4, which are high risk medical device. You need to categorize, right? So that is very important. Like for swab testing, we have different medical devices and all. So all these things you can do. Then PMCF, post-market clinical follow-up is very important. PMCF for medical devices especially. There are different regulatory submissions and all. You can get into more detail as and when. And even FSCA, on-field safety critical assessment. That is the full form is on-field safety corrective actions. So this is very important. FSC assays where you address any particular safety issues, because medical device not necessarily it's external. It can be internal as well, right? Like pacemaker. What is it? It's a medical device. You instill it within the body, right? Amputations. So once there's amputation, you can attach a particular limb. So that is again a medical device. So you need to take into that as well into consideration. Next up, we'll be covering the pharmacy practice. So pharmacy practice, all you need to do is thorough research of the data. So the databases like PubMed, Scopus, Cochrane, Consensus can be useful, and Micromedex you can use on the Play Store as well. From the Play Store you can download. So there are different study designs, like there are different trials that is carried out, right from the basket decentralized trial to the randomized control trials, different studies like cohort, cross control, case studies, and different questionnaires. So there are different questionnaire grading scales, like you have Morinsky's scale for medication adherence. You have different scientists have different kind of scales for patient satisfaction, for patient compliance, for what do you say, medication adherence as well. So the G power is calculated using statistical softwares, and for this you can go for pilot studies for feasibility, reliability of the medication. You can go for the MTM as well, medication therapy management. Then telemedicine also you can have a project upon telemedicine, digital health and all, because nowadays digital therapeutics is also gaining a lot of importance and all. So there are different observational studies, consort studies, stroke studies. You can go for those observational interventional studies. You can go for evidence-based approach as well in the pharmacy practice. You can go for HEOR, health economics, outcome research, pharmacoeconomics, pharmacoepidemiological data. And so these are all the things that you can actually go for while trying. The next subject that we'll be covering is a clinical research. So in clinical research, the literature review again remains the same. You need to perform meta-analysis, network meta-analysis. You can plot volcano plot, forest plot. You can have a CRF, case report forms. You can have a safety efficacy comparison. You can have a salt form comparison. You can have a bioequivalence comparison. You can have a different kinds of DMC reports, and you can perform studies as well on those. Then LPLV is also very important for a study closeout. Last patient, last visit. And then there is first patient, first visit. FP, FV, LP, LV. First patient, first visit, last patient, last visit. So that is also a very important criteria for study close in and study close out. You must be aware. Then there's a RMP, where you have a risk management plan and protocol that you need to follow. And finally, RWE, which is real world data and real world evidence, where you collect data from the actual real world experiences. Then up we have regulatory affairs. Regulatory affairs is nowadays getting a lot of amount of money. Those who are into regulatory affairs, because dossier filing, CTD filing is not easy, especially ECTD, electronic common technical. So if you see M4, M8 guidelines of ICH, multidisciplinary M4, M8 guidelines, it consists of these CTD, ECTDs, where you have PSURs and all update. PSUR is nothing but a periodic safety update reports. So these reports are submitted for evaluation of the risk assessment and all. There's a product life cycle management. And then Six Sigma, basically, that is very important. There is a different models of Six Sigma, DMEDEV model and DMEDAR model. DMEDEV is define, measure, analyze, design, and control, while DMEDEV is define, measure, analyze, design, and verify. Right? Then the different models. Besides this, also the different models, like in the risk management cycle, you have different. If you see that flow chart, risk assessment tools, risk identification, risk hazard analysis. So then there is HACCP, hazard analysis and critical control points. There is FMEA, failure mode effective analysis. And there is FMECA, failure mode effective criticality analysis. These are analysis you can perform for a more detailed idea about the particular regulatory affair, the drug for which you're performing or formulation. So different modules you can target: module 1, 2, 3, 4. Module 1 is administrative, 2 is summary, 3 is the quality data, where CMC, chemistry, manufacturing controls, and you need to identify, you need to even learn about DMF, drug master file, the different parts of DMF that is to be learned by you. Next up, we have the subject of pharmaceutical analysis. So in analysis, basically metabolite profiling, impurity profiling, analytical method validation is all you can do, method development, baseline correction and all. Besides this, there can be options for force degradation studies or accelerated stability studies that you can go for. Right, nowadays, it is having a lot of importance. And analysis might include all of these particular points. Then we have pharmacoinformatics. So there are different tools, like even for biotechnology, if you tell BLAST, FASTA, basic local alignment sequence tool. These are all different tools that are used for primer designing, for different BLAST and all. So you must be aware about these tools that are pharmacoinformatic tools. Then you can set up your experiment design, either vaccine, immunoinformatics. There are so many different terminologies, even pharmacoinformatics, ioinformatics, immunoinformatics, and all that you can go for. You can have different values: RMST, relative mean square deviation, RMSF, relative mean square fluctuation, PSA, polar surface area values. Then you can even incorporate Lipinski's rule of 5, right? So these are different tools. You can incorporate Weber's rule, you can incorporate Ghosh rule. These are different the rules of pharmacoinformatics you must know. Besides this, you can perform the Hammett's steric parameters, Taft's steric parameters, Hammett's electronic parameters, Hans analysis as well. Then you can identify, or if you want to draw the structure, there is Chem Sketch, there is Chem Draw software as well, Chem 3D, on which you can draw a particular structure. And then you can have a molecular computational chemistry is what? Protein-protein interaction, molecular modeling, ligand binding, simulation. So you have a grid size, grid box, and grid size is very important function you must be aware of. You can have a virtual screening, drug likeliness screening as well. And then finally, the last one we have is pharmacology, the field from which I belong. So in vivo, I've already discussed with you all in one of the videos prior, and in silico, I just discussed in the pharmacoinformatics. So this is an in vitro data. So if you want to go for in vitro data, the first thing you need to know is the cell line. So there is which cell line, cell culture, or passages that you're carrying out, what assays you are planning to incorporate in your in vitro, because there are a lot of assays that you can perform, right? So which assay, which enzymatic activity you want to check, and how you want to check, like there is FACS, fluorescence associated cell sorting. If you want to check the protein interaction, there is cell phase plasma resonance. There are so many different techniques that you can actually have you know hands-on practice with. Flow cytometry. Then finally, you need to know about the cell seeding. You need to know about the apoptotic pathways like caspase 9, 8, 1, 3 execute, because cell death pathways are very important for cell viability, cell proliferation studies. And finally, you can have the images for these software, and you can quantify these images using ImageJ. So I have also personally used this software ImageJ for quantification of the PSR staining and the hematoxylin and eosin staining as well. So finally, the data interpretation, where you can have a dose optimization studies being characterized, dose response curves being plotted, and finally publication. So that's it for today's video. I hope it was insightful for you and a multidisciplinary approach you would have got, because any institution, whichever college you work, so like I belong from Naipur Mohali. So here, why are there so many different domains? They are interlinked to each other, right? So you can collaborate anything like cognosy, suticology, chemistry as well. Chemistry से molecule लाओ, cognosy से कोई natural hooks, check the synergistic activity, cognosy and chemistry हो गया. Sutics में formulation बना लो, Cology में check the in vitro toxicity, in vivo toxicity studies. So Cology, Sutics, chemistry, cognosy can be linked in one particular project itself. So it's upon you that how you want to work ahead. Informatics से you can have in silico screening, so आपका पांचवा subject भी add हो गया. Biotech में तो you can have some vectors, you can have some antibodies or mimetics that you can inject via tail vein and all. So biotech भी आ गया उसमें, है ना? Biotech में तो there is again fermentation, and I did not cover because I've previously spoken about biotech a lot, right from the fermentation to the monoclonal antibodies to the vaccine designing to the personalized medicine and all, and different again techniques as well, like RT-PCR, Western blotting and all. So that was suffice. So that's it for today's video. I hope you do share this video, and I know it's a very long video, but I do expect you all कि आप हर aspect पढ़ो सुनो. You will get to learn a lot of new things. Even when I was making this presentation, I got to learn a lot of things in the process. So I hope that it will help you ahead. And ऐसे ही प्यार करती रहिए. Do like, share, subscribe my channel for more such upcoming pharmacy videos.